Hi everyone, welcome back to the channel. This is Dr. Hassi and today's topic is BOT 3303 Module 4 Study Guide. Okay, so in this module we have three chapters, 27, 29, and 8 we are going to cover. So as always, check your PowerPoints, your notes from the class, and then readings obviously from the book. So keep doing deep reading and take notes and review your notes and then look at some of the tips here in this video. Okay, so we start with chapter 27. This is plant hormones or phytohormones. So plant hormones or phytohormones, they promote most of the time and sometimes inhibit certain physiological activities. So they are also called phytohormones, plant hormones. So we have 5 major hormones, and we have a few newly discovered hormones in this under this chapter, like auxin, cytokinin, ethylene, ABA, gibberellins, 5 big ones, and also we have some other ones like salicylic acid, propranolol, steroids, and also jasminic acids that newly discovered. Okay, so let's talk about each of them. Now, they all involve in signaling. You can see in this table and differentiation like gene expression. They all influence gene expression, but they might be influencing also rate of cell division or cell expansion, depending on which hormone. Right? So auxin is our #1 hormone, like IAAs. They are produced should a meristem, and they influence growth, flowering. So make sure you review about their functions and their location and their translocation places, right? So they could inhibit stuff. Also learn about what they could inhibit, and then they come from like a precursors like tryptophan, right? So cytokinin is our #2 plant hormone, which is again another growth promoter like auxin, but in this case they are not producing shoot, but they're producing roots, and then they move up to the shoot, and then they involve in these physiological functions like growth and flowering. A lot of similarities with auxins. They go together a lot of time, but obviously they stimulate cell division, cytokinin. So the name suggests they are involving cytokinesis. They could also delay senescence. That means early death of the plants. They could involve in growth of the shoot. So there are experiments and some research also we cover. So make sure you learn about this. Also, you learn about auxin cytokinin ratio. So not just individual effects, but as like how they interact with each other and their ratio influence plant growth. So our next hormone is gibberellins. It's another big hormone we see in shoot apical meristem and root apical meristem. We see it's involved in internode elongation, so enzyme mobilization during the germination, fruit ripening, abscission, and a lot of other germination-related topics involved with this hormone, like breaking dormancy, so and causing taller plants. So read about this one, and next one is ethylene, which another very unique one, which basically precursors of methionine. So we learn about ethylene is involved also in stem elongation. So we learn about some experiments done on this and fruit ripening. ABA is a very unique hormone. You can see in this table that it's actually its growth inhibitor, like similar way with ethylene and ABA. They both inhibitors. So ABA, you can see in mature leaves, fruits, they are suppressing stomata closing, a lot of physiological involvement of ABA, so you can learn about. And then salicylic acid, propranolols, and jasminic acids. So please read about those too. So our next chapter is plant genetics, which is chapter 8. There is a lot of vocabulary in this chapter, so it's better to start with getting down the vocabulary, learn about these basic vocabulary like diploid, haploid, dominant, recessive, egg, fertilization, gamete, genetic recombination, genetics, genotype, phenotype, haploid, heredity, heterozygous, homologous, homozygous, hybrid, and different generations. So we have character and trait and gene. They are interchangeably used. Character is heritable feature like fur color, but trait is the variant of the character, right? So like brown or any other colors. These are traits. Mendel did 7 year work on different traits in pea plants as his model plant from 1856 to the 1863. And he published his work in 1865, and he's considered father of genetics. He bred thousands of pea plants in the 7 years, many traits controlled by one gene or more than one genes. So he found, and then gene passed from one generation to the next generation. Also, he found, and also there is something called incomplete dominance when blended snapdragon petal color we see like pink color. Um, so in plants, so since genetics started with plants, Mendel worked on plants. So plant genetics is very, very important. So make sure you learn about monohybrid, dihybrid, QTLs in plants. And also we have exact same linkage groups and linkage maps in plants. Like we have many linkage maps of plants actually completed already, like tomato, Arabidopsis, maize, some of the plants. And also meiosis also involved here in the reproduction in plants. We have parental generation, the parental gametes, and then F1 genotypes. Again, so complete genetics of plants. So make sure you review everything in this chapter 8 involving with all this vocabulary. Chapter 29 is our next chapter, a really fun chapter, which is plant nutrition and soils. So plant nutrition and soils involve with essential nutrients. It means they are essential. That means they are not beneficial only, but they are essential. That means they cannot be replaced. They must be present in order to really complete the life cycle. They are essential, right? Also, soils are part of this chapter. So we have 17 essential nutrients or elements. 9 of them are the large macro group, which are nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, hydrogen, oxygen, and again nitrogen, which freely available in the air, but nitrogen is one that's specific that plants cannot really use as in the form in the air. So it needs some help. It needs to be converted to form plants like to use it. Okay, so phosphorus, very big one, and make sure that you learn about these different deficiency symptoms, how plants tell. So they are actually missing certain nutrients. They show a symptom actually in young or old part of the plant so that we can actually detect and we can actually find out what nutrients is missing. Okay, again, macro versus micro. And then different soil types like sandy soils, loam soils, silt, peat, clay, different types of soil and soil levels like A, B, C, O, and R horizons that make sure and you actually review. And erosion, like loss of topsoil, very critical. So soil takes up to 100 years or maybe so to develop. So if we actually lose the soil, it's a big loss. So it takes very long time to replace lost soil. So keep reading on these 3 chapters, read your notes, and I will see you next time in the class. Thanks for watching. Please feel free to connect and subscribe to the channel. See you next time and happy studying.